Well, my lab basically works on a new type of therapy of cancer, which is called liposomes. Okay, you guys don't need to copy this. Okay, so liposomes are basically a sphere of lipids which hold cancer drugs inside it, which are these, and it's made out of lipids which cause the drugs to not leak out, and they're specified to go into cancer cells rather than just like bind onto healthy cells. And the polymers called polyethylene glycol are attached to the liposomes, which help them flow in the bloodstream better. And it's also attached with antibodies. Okay. So the way they get into the cancer cells, they bind to the antigens. These are the antigens with the antibodies that are attached on the liposomes. And then there are various ways they go inside the cells. This is the ideal path where they go in through the endosomal path through endocytosis, and then they go into the endosome and they release the drug this way. But then they could also go into the lysosome where they just dissolve and nothing happens. Okay. So in this experiment, we were basically comparing liposomes, regular liposomes which had no antibodies on it, and immunoliposomes which had antibodies, which were specified to go to the cancer cells, and we were using human ovarian cancer cells. And the antibodies that were used were called Herceptin, and it was attached to the polyethylene glycol, which was called PEG, and the regular liposomes were used as our control. And we hypothesized that the immunoliposomes would bind and internalize better inside the cancer cells, which was basically, it would release drugs more inside the cancer cells than the regular liposomes. Okay, so these are the materials. Those are the liposomes. It shows fluorescence with the drugs inside it. Those are the trastuzumab antibodies. That's the extruder, which we use to make the liposome smaller so it's easier to go internalize into the cancer cells. And that's the spectrophotometer, which it shows fluorescence when it is actually attached to the cancer cells. And these are all the materials. Okay, so the method, we basically prepared the liposomes first with the PEG and table. Okay, so this is basically the data table, and HPTS was the fluorophores, which basically emit the fluorescence in the spectrophotometer. And we found that these are the regular liposomes, and these are the immunoliposomes. So we found that the immunoliposomes released more drug and attached more onto the cancer cells than the regular liposomes. Okay, and another one is, this is the calibration chart where we try to find the pH of, okay, so when liposomes go inside a cancer cells, there's different pHs. So when the liposomes are flowing inside the bloodstream, the pH is 7.4. But when it goes inside the cancer cell, the pH drops. So this is the data that we collected, and you see the time over here, so this summer we are going to be working on a different type of liposome called DSPA. We used to be working on DSPC, which was already a studied liposome. So our lab, my professor and the lab student, they proposed DSPA, saying that it is more pH sensitive and it releases more drugs as when it goes inside the cancer cells as compared to DSPA when it goes inside the cancer cells. So these are drugs that they have collected like in March, and it shows that DSPA, the pH lowers, which shows it is more pH sensitive, and it releases the drug more as compared to DSPC, which did not release as much drug. Okay, so that's basically it. I would like to thank Professor Sofu, Sharon Carvin, my co-mentor, everybody else in the lab, Polytech University, Miss Narain, Dr. Stat, ACS staff, Harlem Student Society, and family and friends and everybody.
The only way to know if the liposome is in the cancer cell? If the pH difference is the only thing that you can know?
If it's inside the cancer cell, yeah, because I don't think it's the only way, but that's how we find it in the. Okay, that's how we find it in the lab, because we use spectrometry, which shows the intensities and well, you know, by knowing the pH level, you know that inside the pH level is a lower pH level, so that's how you know that the drug released inside. And the drug, like, you mix it with fluorophores inside in the lab, so you know that it actually went inside. But I don't know if there's any other way. We hypothesized that the immunoliposomes would attach more and bind more onto the cancer cells than the regular liposomes.
How does the drug kill the?
How does the drug go into the cancer cell?
How does the drug kill the? How does that happen?
Well, it's a drug to kill the cancer. I don't know if they're studying on that, but we just study on how the liposomes went inside the cancer cells and how to make it better, so it could like release the drug inside the cancer cells.
But that's a good question. You should ask him and should ask.
Okay.